welcome our guest speaker here today, Linn Hoffmann. So, Linn is a lecturer in marine botany in the Botany department. Linn did her PhD at Karl University in Germany, and once completing that, she did postdoctoral stints in Gothenburg in Sweden, and also here in the chemistry department at Otago. In 2013, she led a Emmy Nova research group at Kiel, and in 2014 she returned here to Otago to take up a faculty position in the Botany department. Oh. Linn's work concentrates on the implications of environmental stresses and on marine phytoplankton, ecology and physiology. Linn has a special interest in the effects of ocean acidification and the trace metals from volcanic eruptions and the effect that they have on marine phytoplankton communities. So without further ado, I'd like to welcome Linn to the seminar, and we'll leave it to you. Kia ora.
I have this one, so probably you want to turn that on. Thank you. Welcome, everyone. So the title of my talk is The Blue Lungs of the Planet, which really is what phytoplankton, it really describes well what phytoplankton does and the role that they play on our planet. So primary production in a changing ocean. And I thought long and hard about just click on the slide. Should work now. Okay. Yeah. Okay. So I thought long and hard about what I wanted to include here, and initially I had a list of probably 6 or 7 things, and then I had to narrow it down more and more. So I decided that I'm going to talk about ocean warming and deoxygenation, and then of course ocean acidification, and in the end I want to talk about microplastics and how they impact, well, I'm just going to really briefly talk about how they might impact phytoplankton. Of course, there's a much broader impact of microplastics. So there are lots of other things that could have been included here, but as I said, you have to start somewhere. So first of all, I want to give a little introduction. Not everyone might be very familiar with phytoplankton, so I just want to show some beautiful pictures because I like them so much, and just explain what phytoplankton actually is and why they are so important. So usually when you think about primary production in aquatic systems, you think about macroalgae. But macroalgae are only really we only find them at coastal areas. So in the open ocean, we don't have macroalgae, except for, if I have time, I'll mention sargassum in the very end, which is an exception to that. But let's just go with this. So in the open water column, in coastal and in open ocean waters and in fresh water, we have these beautiful creatures. So phytoplankton. They're unicellular algae and also cyanobacteria. Cyanobacteria, sometimes called blue-green algae, but they are not algae, they are bacteria, but we also count them as phytoplankton because they photosynthesize. They are all really, really small. Some of the species stick together in these longer cell chains, or these aggregate balls here. So those aggregates can be bigger. The colonies can be bigger, not aggregates. Colonies, they can be larger, so up to several centimeters actually, and we can see them. But the individual organism, the individual cell is always really small. Some of them can swim a little bit, but they're all unable to move against currents. So they're more or less passively floating in the water. Yeah, just some beautiful pictures. So we have some calcifying ones, this one here, for example, or this one here, the coccolithophores. I particularly like this one. It looks like little muffin cups that it's covering itself with. Others, the diatoms here, they have a silica frustule, so they built their own little glass house that they sit in. Others look fairly boring little green blobs, but some of them have really sophisticated forms and shapes. And I remember well the first time as an undergraduate student that I looked through a microscope, and we were looking at diatoms in Kiel and the Kiel Bight. And it's just amazing when you look at a drop of water under the microscope what you can find there. And I immediately knew that I really wanted to work with them. So this graph, or this slide here shows the average global sea surface chlorophyll concentration. So it's averaged over several years. And what we see is that the highest chlorophyll concentrations are in coastal regions, so close to the coast, which is where we find most of the nutrients. And also, of course, there is macroalgae growing there as well. But all this chlorophyll here in the open ocean, so all of this, that's phytoplankton and just phytoplankton. And so when you think about the open ocean as the largest biome that we have on this planet, even though phytoplankton organisms are really tiny, they can sometimes grow very quickly and reach really high biomasses. And because we have this really large area of the open ocean, they account for 95% of the total marine primary production. And when you take all the land plants into this equation as well, phytoplankton account for about 50% of the global primary production. So this is why we talk about the blue lungs of the planet. It's not just the rainforests, of course they are important too, but phytoplankton is really critical for primary production and oxygen production. So I always tell my students, think about this in terms of every second breath you take contains oxygen that was produced by phytoplankton. So that really clarifies how critical it is. And you can imagine that every significant change in phytoplankton productivity, but also species composition, might have a really, really significant impact on marine systems, but also on global systems. Okay, so let's talk a little bit more about these strange creatures floating in the ocean and how they manage to do that, and what problems they are facing when they do this. Sorry? Okay. On this side. So they are, phytoplankton can only grow in the surface waters when there's enough light for them to photosynthesize. So all the deep waters that are too dark, phytoplankton can't grow there. Now, luckily, we have something called stratification in water bodies. So the surface is warmed up by the sun, and also you have fresh water coming in. So you have a less dense surface water body that's sitting on this colder, more saline deep water. So we have a density barrier between the two. And that means that phytoplankton aren't constantly mixed down to very deep waters. So they, most of them can stay in surface waters. They can also regulate their buoyancy a little bit. So as long as they are buoyant, they wouldn't just sink out into deep waters. That means that in the surface waters where there's enough sunlight, they photosynthesize and they take up nutrients. So the nutrient concentrations decrease in surface waters. Now, when they die, they either get eaten by zooplankton and fish eventually, or they stick together. When they die, they become quite sticky. So they form these larger aggregates. You might have heard of marine snow, which is basically what this is. So these larger aggregates that are then very dense, can't regulate their buoyancy anymore, and they sink out into these deeper, darker waters. Down here, we have remineralization, most of the remineralization happening. So bacteria remineralize these organic nutrients, and that means we end up with an increase in the nutrient concentrations in these deep waters. The way I like to think about is that the deep ocean is kind of like the soil and terrestrial ecosystem. So that's where all the nutrients sit, where you don't have light for photosynthesis. Now, plants are very lucky. They have their tree trunks and the roots to move the nutrients up to where all the light is, where they can photosynthesize. Phytoplankton can't do that. So they need to rely on deep water being upwelled or being mixed to the surface. So they really need these mixing events and diffusion of nutrients through this thermocline into the upper water body where they can use the nutrients. They also need to rely on rapid nutrient recycling in surface waters by bacteria. So that's another really critical aspect. So some bacteria are also obviously also up here, and they need to remineralize the nutrients before everything falls out. Otherwise, there wouldn't be enough in the surface waters. Right. So let's talk about ocean warming, and I want to talk about deoxygenation at the same time because they kind of go hand in hand, and I didn't want to show lots of pictures of dead fish, so I just choose this cartoon instead. So what's happening when the ocean warms up? Well, lots of different things happen in different regions at the same time. So I'm trying to simplify it by looking at the main differences in low latitudes and high latitudes. So at low latitudes, we already have quite a strong stratification. So in tropical regions, the surface waters are already very warm. We have a very strong density difference between surface and deep waters. That means we have relatively little nutrients being mixed up to the surface ocean. If this increases and keeps going, we will have even more stronger stratification. So the nutrient input to surface waters will decrease even further. So we're expecting to see less phytoplankton grow and more severe nutrient limitation in those regions. In high latitudes, the situation is a little bit different because the surface waters are colder, and because of that, we don't have this, we have a density difference, but it's not quite as strong, and it's also more seasonal. So during winter months, for example, we have a lot more mixing because the surface waters are much colder, so there's less density difference. So on average, we have a lot more nutrients coming into the surface waters. But because of this deeper mixing, phytoplankton in high latitudes is more likely to be light limited. So they're mixed to deeper, darker waters. Now with ocean warming, we will see a stronger stratification and also a shallowing of this stratification. So the mixed layer, the mixed surface layer will become a little shallower. That means that on the one hand, we will have less nutrients being mixed up, but because the phytoplankton here are mainly light limited, they might be able to benefit from the increased light intensities. So on average, we're expecting higher phytoplankton growth in high latitudes. Okay, so higher phytoplankton growth, that sounds like a good thing, right? We want more primary producers to grow and take up CO2. I'll come back to that. First of all, warming, and I want to mention the importance of time series and some of the work that my PhD student Morgan Myers, she just finished or just defended her thesis last week. She did some work on the zooplankton distribution in New Zealand waters. So she was working on the Munida transect with Kim. And what I want to stress is the importance of time series studies, not only for the background data, but also for the biology in basically everywhere, because otherwise we can never tell if we see differences, if climate change is already impacting ecosystems, and what our baseline data would be like. So in this region that we're talking about here, we only have data from that were published in 1976 by Gillett. I think those data were actually taken in the 60s. Then there's a study published in 2016 from Blue Skin Bay, and then there's Morgan's data from the year 2017 and 18, and that's it. So we can't really say much. So I really want to emphasize how important it is that we do more regular sampling here. And zooplankton is not what I usually work with, but because they directly feed on phytoplankton, their distribution and biomass obviously has really significant impacts on the standing stock of phytoplankton communities. So the question was, do we see differences here? And also coincidentally, during Morgan's sampling time, we had this heat wave event. So what Morgan found is the data here, they show the temperature difference. So the dotted line are the temperatures that were reported by Gillett et al., so taken in 1966 and 67, and the solid lines are Morgan's data. And you can see that especially in January when we had this heat wave event, there was a huge difference. Again, these are just snapshots. Of course, we have more temperature data than that, so it's, when we want to put that together with the biological impacts, we only have those data to go with. So you can see the temperature was always slightly higher, might not be significant, but definitely in January we had this massive heat wave event. What Morgan noticed was that in the Gillett et al. Study, the dominant salp species was Ilea magalhanica. So that's a sub-Antarctic species, so that likes cold waters, and that was really dominant in these along the Munida transect. Instead, during Morgan's study, she didn't find Ilea magalhanica, but she found Thalia democratica, which is a more warm water species. again, we can't really say much about those just 2 data points. She especially found it in March, so right after the heat wave event. So, I mean, we don't know if those 2 species maybe appear in the same waters at different times of the year all the time, or, you know, if this is just annual variability, or if this is really an indicator for a broader shift in zooplankton grazer composition. And especially these salps here, when it comes to zooplankton grazers, they are like the vacuum cleaners of the ocean. So large salp appearances can really diminish phytoplankton blooms very, very quickly and really determine the phytoplankton community structure very significantly. So that was interesting, but again, I want to stress we really need to invest more effort and time and money into these time series transects. Otherwise, we will never be able to tell what's changing and what not. Unfortunately, time series don't really give you Nature papers, at least not quickly. So it's a lot of work, especially these counting these biological specimens and identifying them. All right. So going back to the phytoplankton, more phytoplankton is thought to be a good thing, right? Because they take up CO2, they photosynthesize, they reduce CO2 concentrations. Well, too much of a good thing is almost always a bad thing. And that's the same with phytoplankton biomass. So we especially know this phenomena of oxygen minimum zones from coastal regions. When we have phytoplankton growing, as I said, the dead organic material sinks to deeper waters, and the bacteria that remineralize it, they use oxygen when doing that. And because we don't have photosynthesis happening in deep waters, we don't have any oxygen being replaced there. So the only way to replace the oxygen down here would be mixing with higher oxygen waters. If we have a lot more biomass, a lot more sinks out, a lot more oxygen is used up, and we end up with these dead zones. That's a phenomenon that's especially known for coastal regions where we have the impact of eutrophication, but we also have that in the open ocean. So it can really kill fish and can kill large amounts of fish relatively quickly. Obviously, different fish species have different oxygen requirements. Fish are usually, if they can swim away, they can do that. But of course, you also have sessile organisms that can't move away. So then if their oxygen concentrations get too low for them, they'll just die. As I said, this is mainly known for coastal regions, but it's also happening in the open ocean. And it's not, it's a natural phenomena. So we always have lower oxygen concentrations in these mid regions where we have the bacterial respiration happening. And it doesn't necessarily mean that this is killing everything. It really depends on what the actual oxygen concentration in those waters are. We are also expecting to see an increase in the extent and also the frequency of those low oxygen events and of those oxygen minimum zones. But the difference, but it's slightly different processes for open ocean and coastal regions. For coastal regions, it's mainly eutrophication that's driving this. We have all the agricultural runoff, especially in coastal systems, that is mainly driving this mass growth of phytoplankton and other algae in coastal regions and leaving these dead zones behind. In the open ocean, it's also a combination of warming. So warm waters can hold less oxygen. So if as seawater warms up, it can hold less oxygen. So that means just changing the temperature changes the oxygen concentrations. So that's why these open ocean oxygen minimum zones will also start to expand. And then in the regions of the open ocean where we have upwelling, where we have those high nutrient waters being brought to the surface, that will also cause more phytoplankton to grow and to sink out. So we're expecting to see large changes in these low oxygen conditions. And that's not only killing fish or all kinds of other animals, but it's also really changing the whole energy flow through food webs. So when we think about, we call it the biological pump, so the flow of carbon from the surface ocean to the deep ocean. When we think about that in oxygenated waters, some of the biomass, of the dead biomass that's produced in the surface sinks to deeper waters, but also some of it is moved up to those mobile predators. So it's moving up the food chain. And some of it is remineralized by bacteria, and another part here is then used by benthic organisms. Now, when we have these oxygen minimum zones and our larger predators have to leave, so this is for the open ocean where we only have these oxygen minimum zones in the mid layers, that means that we have less energy going to higher trophic levels. So that means more energy reaches the seafloor and more is used by those benthic organisms at the bottom of the seafloor. If we have the same situation in coastal systems where we usually have the dead zone sitting right on the seafloor and where we kill all those benthic organisms as well, then all of the energy goes to bacteria. So we really have a decoupling of the carbon flow through food webs away from those larger mobile predators to, first of all, the benthic organisms, and in extreme cases, just to bacteria. Okay, so I mentioned ocean warming and how it's impacting temperature and mixing. It can also, of course, have larger scale impacts on ocean circulation. And this is all caused by changes in the climate caused by atmospheric CO2. At the same time, CO2 does something different. It's impacting the ocean chemistry through ocean acidification. So I want to stress that this is a different process. So ocean acidification is not the same as climate change. Both of them are caused by CO2, but it's different processes. All of these things together work on organisms in the pelagic, so in the open water body, and all of them will eventually impact carbon sinks and sources in the ocean, which then again has impacts on climate, on atmospheric CO2 and our climate as well. So it's a complex system. So let's have a look at ocean acidification. I'm not going to go into detail about what ocean acidification is. If someone's not familiar with this and has questions, I'm more than happy to answer them afterwards. But for now, I'm just going to say that as we have more and more CO2 in the atmosphere, there's always gas exchange between the ocean and the atmosphere. More CO2 enters the ocean, and that is changing the carbonate chemistry of the ocean, and it's changing the pH. What we see from long-term monitoring is that the black line here is the atmospheric CO2 concentrations, and the colored lines are the PCO2 concentrations in the waters at these different monitoring stations. And down here, we have the in situ pH in the water. So we see as atmospheric and seawater pH rises, on average, we see a decrease in pH. Now you will notice that most of those monitoring stations are in the Northern Hemisphere. And we're really lucky down here to have Kim, who's doing amazing work monitoring seawater carbonate chemistry on the Munida transect since 1998, is it? So these are her data. What we see here, the red line is the atmospheric CO2 concentrations from Bering Head on the North Island, and those light blue and dark blue points here, those are, the dark blue are the seawater PCO2 concentrations, and the light blue ones are seawater pH. So we see the exact same trend down here. I put the those two Northern Hemisphere and Southern Hemisphere figures here because I have been asked by people, yeah, but most of the carbon emissions happen on the Northern Hemisphere and it can't possibly impact us down here. So we see the exact same trends down here as well. But on average, we have a lot less of those monitoring stations in the Southern Hemisphere. So that's a real problem for understanding those long-term trends. But what we do know from the data that we have is that the ocean is acidifying. We already have seen a drop of about 0.1 pH units, which doesn't sound much, but because it's a logarithmic scale, it is actually quite a lot. Compared to pre-industrial times, and depending on how we go with our carbon emissions, and it's not looking good, we will see a lot more drastic decreases in seawater pH in the future. And I want to put this into context because seawater pH, of course, has been different over geological times. But roughly during the last about 20 million years, seawater pH has always been above 8. So this is really what the marine ecosystem has evolved to live with. And everything that goes below 8 is there we start to see dramatic changes in the ecosystem, in the response of different individuals, different species. And what we see here, what we're expecting until the end of this century, is such a drastic decrease in seawater pH, which we have never seen before. So changes in pH per se are not really so much of a problem if the ecosystem has enough time to adapt to it. But we're not giving them that time. So the changes are happening so rapidly that organisms just won't be able to deal with it. All right, but CO2 is a good thing, right? Phytoplankton and macroalgae, just as plants, need CO2 to photosynthesize. So what's the fuss about more CO2 in the ocean, even though it might be impacting the pH? Well, not all algae are doing well under low pH, so under high CO2 conditions. Some of them are calcifying, so they form these little calcium carbonate scales, the so-called coccolith. And those coccoliths under low pH start to deform. So what happens is, because we have a change in the carbonate chemistry of the seawater with more CO2, calcium carbonate starts to dissolve. And it's more difficult for calcifying organisms to produce new calcium carbonates. And they need to invest more energy to do that. Those are just calcifying phytoplankton. Most calcifying organisms are not phytoplankton, so we're talking about mollusks and corals and the like. A really interesting or good way to study the impact of ocean acidification, well, we can do lab experiments, of course, but if we want to look at larger ecosystems, what we can do is use those volcanic CO2 seeps. So those are regions where, because of volcanic activity, we have CO2 bubbling out of the seafloor. So we have kind of a natural ocean acidification laboratory, if you will. There are several sites all around the globe where this is happening, probably a lot more, but those are the ones that that people have worked on. I put New Zealand here. This is Whakaari White Island. We don't only have CO2 bubbling out there. There's a lot of other metals and acids coming from that volcano. We did a study there as well. I'm not going to show those data. What I want to concentrate on are data from Japan, where one of my PhD students worked on a few years ago. So this is a small island off the coast of Japan, Shikinejima. And you see in this little bay here, we have this very localized area where the CO2 is bubbling out of the seafloor. And you see this massive increase in PCO2. So remember, at the end of the century, we're expecting a PCO2 of roughly 1,000. So we have some stations that go well beyond that, but we have a large area where we have that we could work with. Our control site is just a few hundred meters away, just the next little bay. So it has the exact same environmental conditions, temperature, light, tidal wave exposure, all those kind of things. So it's really the ideal control station. What we can see in not only these systems in Japan, but all the other CO2 vents that have been studied, is that as we go from, so here on the left-hand side, we have our control at 300 microatmosphere CO2, where we have lots of calcifying organisms in the intertidal here, and also in the subtidal, we have beautiful coral reefs. And then as you move up, as you move to 400 and then 1,000, in this case, 1,100 microatmosphere atmosphere CO2, you see how these communities change completely. So the calcifying organisms disappear, they can't calcify anymore, or they can't settle there. And we see that these so-called turf algae take over. So those smaller algae that really take over, because they often can make use of the increased CO2 and photosynthesize better. And I wanted to include this picture because one thing that's quite different in Shikinejima compared to the other CO2 vents is that we have everything at these high CO2 stations covered by these thick mats. And when this is Ro, one of my former PhD students, he went up to Japan in 2017, I think 16, 17 probably. So when he went diving there and saw this, he sent me this picture and he said, Linn, there's a marine station there and lots of great people working there. And he said, they think it's a diatom mat. And I said, there's no way that's a diatom. So diatoms are phytoplankton organisms. They can form long chains, but I said, this is no way that's a diatom. So he ripped off a little piece, put it under the microscope, and it turns out they're absolutely right. It's Bidulphia bidulphiana, which is a diatom that has these long chains that it forms, and they're quite sticky, so that chains stick together. And there are some reports in the literature that it can form mats, but I've never seen anything like this or heard of anything like this. I also want to mention this little snail here. You can see how polished it is, so we can really see the calcium carbonate dissolution in this area. And these mats, it's really like this, how do you call this, lawn that you can buy, that you can roll? It's really like this. You can rip off huge pieces. Everything is anoxic. It's completely smothered underneath. There are lots of invertebrates living in it, though, so it's not completely dead. But it's, of course, a completely different habitat compared to just a few hundred meters around the corner, where we have this coral reefs. We don't quite know why we have this mass appearance of this particular species there. So it's something that we have a paper in review at the moment, something we are hoping to find a bit more answers for in the future. But what Ro actually did there, when he was working in Japan, he was interested at biofilms. So bacteria and phytoplankton, some phytoplankton don't float in the water, they grow on surfaces. So he wanted to know how, if we see differences in the settlement or in the establishment of biofilms. So he put out little plates, lots of them, and looked at, here we have the controls, and here we have our low pH stations. And it's a bit difficult to see. I hope you don't get a headache from all the colors. So we have day 5, 10, 15, and 21. And for each day, we have 7 replicates. This is just the division on the divisional level here, what we see for the eukaryotic community. So what I want to stress out is that in the beginning at day 5, we have quite a different community compared to all the rest of our sampling days for both the control and our high CO2 concentration or stations. And then after day 10, it's quite variable, but on average, we don't really see a clear trend. So it seems to have settled down a little bit. But what we do see, we see a significant, and Ro obviously did all the proper statistics on this, so there's a significant difference in the community composition between our two sites. So seawater pH does impact the community composition on those biofilms. If we go down a little deeper and just look at the 20 most abundant genera or species in those two stations. This is a heat map. You might not be familiar how to read it, so I'll explain it. So the darker the green, the more abundant this particular species is. Again, we have the first 7 dots are day 5, and then over time up to day 21. What we do see is that when you look at the second one from the top, Primnesium, so those this genus becomes really abundant after day 10, both in the reference and in the high CO2. The difference is that in the high CO2, it is the most abundant genus after day 2. In our control station, we have other ones that are more dominant. Now, this is particularly interesting because Primnesium is a genus that has a lot of toxic algae. And this has been reported before from completely different CO2 vents, that we see an increase in those toxic Primnesium species under high CO2 conditions. So that's something to potentially watch out for. Another student, Nadjesta, who just started a postdoc in Norway last week. She did her PhD here in Portobello with Miles, and I was lucky enough to be a co-supervisor on this particular project, where she was also working with biofilms, but she was doing long-term incubations up to 14 months. And she was working with ambient pH, just with the Portobello seawater here on the right-hand side. And then she went all the way down to pH 7.0. So she really wanted to push it beyond what we're expecting to see until the end of the century. And you can just by looking at it, see how incredibly different those biofilms are. So there is a huge difference also within this expected ocean acidification range down to 7.7 here. And then beyond that, it's even more. So these red ones that we see on the ambient pH slides, those are crustose coralline algae. So those disappear completely at lower pH because they are calcifying. And then completely different communities taking over. So it's not only in the first few days like what Ro was working on, but also the long-term biofilms look very different. What Nadjesta was actually interested in, she wanted to know if the difference in biofilm community composition had an impact on the settlement of she tested different species throughout her PhD for this study. It was, I forgot the name of this little tube worm here. So, and what she did see, so yeah, here, the white ones is the settlement on 7-month-old biofilms, gray is 10-month-old biofilms, and black is 14-month-old biofilms. So what we do see when we go from a little counterintuitive, when we go from left to right here, from ambient pH down to 7.7, so our expected ocean acidification range, we see a decreasing trend in settlement. But then interestingly, at lower pH, it starts to increase again. At 7.0, there was absolutely no settlement detectable. So obviously that is something that's really harming those organisms. We don't quite understand what's causing the increase here again. So that's really interesting. Apparently these types of biofilms are beneficial for settlement again, but over this ocean acidification range, the trend is quite obvious and clear. I want to quickly mention the variance in seawater pH, because that is something that people often comment on. And they say, well, we have a large natural variance depending on which system you are in. Because, for example, in kelp forests, when you have a lot of CO2 taken up during the day when they photosynthesize, so you have actually a large increase in seawater pH, and then the opposite is happening during the night when organisms take up oxygen release CO2. So people sometimes say, well, when we have this natural variance and it's larger than what you're expecting to see in terms of change in pH until the end of the century, why do we even bother? Some regions, like these are the CO2 vents here, they have massive changes. I mean, CO2 can easily go down to 7 from above 8. The open ocean is much more stable, but then also coral reefs have quite some variability. The analogy that I use is usually, think about this fluctuation here as the money in your bank account. And that's something that people usually can relate to quite well. As long as you're above this critical threshold, this red line here, which is 0 in your bank account, you're okay, you can still eat. You might have one night where you don't have money, but you can, even if you don't eat one night, you can survive that. But then if something happens, if you have an additional cost, the whole thing shifts, so your whole fluctuation in your bank account shifts down, and all of a sudden you are below this critical value much more commonly. And that was starting to impact you. That's the same for those organisms. Variance in itself is not a problem as long as it doesn't go below a certain threshold. And even a small change in that baseline can be really critical, especially for organisms that have very vulnerable stages like larvae stages, or where the reproductive cycle is really, really critical and really impacted by pH. Okay, and I also wanted to mention the Ocean Acidification Action Plan. A lot of countries have started to really realize that this is a problem for especially aquaculture, but also in general for the ecosystem. So more and more countries are starting to release these action plans. This year is the one from California, and I'm part of a group of people from all over the country that work with the government on developing a New Zealand Ocean Acidification Action Plan. At the moment, that is with the Ministry for the Environment, and apparently very, very close to being released. So hopefully we will see that come out soon. Okay. Now something completely different. It has nothing really to do with climate change, but of course it's caused by us as well. So I want to talk about microplastics. And people usually think microplastics is a problem for heterotrophic organisms. Filter feeders that are clogged, or larger plastic particles are a problem for marine mammals and birds. But this really small microplastics can also have a huge negative impact. There are more and more studies coming out looking at the impact of really, really small particles on algae and other organisms. So in this study here, what they have found is that when you add microplastics to a happily growing algae, they stick to the surface. And they are especially, as I said, a lot of algae are quite sticky, and those small plastic particles are especially reactive and like to stick to stuff. So a lot of them stick to the surface of those algae. That in itself causes a decrease in the light intensity that can reach the algae. But it also causes oxidative stress. So obviously the algae can sense that there is something. It's sometimes, I mean, we're not quite sure if it's just the plastic or the particle itself. There are a lot of plasticizers that are released by plastics. Organic compounds, toxic metals can bind to plastic. So that might have something to do with it as well. So we can, in this study here, they found that they found a significant increase in the oxidative stress response, but also they saw morphological changes of the algae. They were growing slower, and you can see that, for example, here the cell membrane was detaching from the cell wall, and they also found a thickening of the cell wall, which seemed to be some kind of protective mechanism, because after a while, the cell was able to counteract those negative impacts and to somehow fight that off and start to grow better again and change the morphology back to close to the normal level. But of course, that costs a lot of energy. So what the long-term impact is, we don't quite know. So there are impacts even on algae and on really small phytoplankton. We have a master's student in the botany department who's working on microplastics in ulva, and she's just dipping the sea lettuce ulva in this microplastic solution, and those are fluorescent particles, so you can make them visible under fluorescent light. And she's trying to wash them off, and I can tell you it's impossible to wash them off. They just stick to the ulva. So she also did stress response markers and is still working through those data. This is a study here led by my former PhD supervisor, Ilka Peken. She was looking at microplastics in Arctic sea ice. And what they found is that they had a extremely high concentration. So concentrations in the same size range as we would expect for extremely polluted coastal regions. And what I find really interesting is here, when you look at the size distribution, this is in micrometers. Most of the particles were found in the smallest size class. Now, this stops here at 11 micrometers because that's the detection limit of the best method that we have available at the moment. But if you see this, I'm pretty sure it will just go up and up and up. So just because we can't detect these extremely small ones unless they are fluorescent in the lab, we don't know what the actual number is of those extremely small ones, but it's much likely, very likely that it will increase because every particle eventually breaks down. In the ice, we not only have plastic, but we also have algae in these little brine pockets, really beautiful ice algae. To hurry up to show these last figures. So one study that Ilka and I did together, and we just published this year, is we were looking at if ice algae and microplastics interfere in sea ice. Our initial idea was that the plastic would bind to the sticky algae, and ice algae are particularly sticky. And then those ice algae, they can move a little bit, and they actually move deeper into the ice. So we thought that they would be a vector of transporting microplastics deeper into the ice. We didn't find that, but we did observe that we had significantly less algae in the ice when microplastics were present. So that was one thing that's interesting. And then we also observed a really strong impact of salinity on the behavior of microplastics, where they were and what they were sticking to. So in the water, when we had higher salinities, as you have ice forming, the seawater becomes more saline around it. So that decreased the relative amount of beads in the water. So they seem to become more reactive and sticking to something. In the ice, we found more microplastic beads when the salinity was higher. So apparently they were then sticking to the ice itself. And also the presence of algae really impacted how many of those microplastics were sticking to our container walls. Obviously in experiments, you always have the container walls themselves, which are a bit of a problem. So stuff is sticking to them as well. Okay, just very, very quickly, because I want to mention this. I said in the beginning that in the open ocean, we only have phytoplankton. That's not quite true. We also have this beautiful macroalgae sargassum, which is natural in, as the name suggests, the Sargasso Sea. It's a wonderful habitat for turtles and fish and all kinds of things, unless you have too much of it. As I said, too much of a good thing is hardly ever good. Since 2011, we see this unbelievable increase in sargassum biomass. Down here, you see the mean coverage in square kilometers. Before 2011, you weren't even able really to plot anything on this scale. Since then, except for 2013, we see this unbelievable increase. And the last 2 years here just seem to follow that trend. And I just want to show this satellite image here. I mean, I just think it's so unbelievable. It looks like you can walk across the Atlantic. So what we think is happening is it's nutrients being released from the Amazon River that are fertilizing this. As I said, it's natural in these environments, but not in this extension. A small part of me thinks, yeah, you think about all this terrible fires that we have everywhere destroying the landmass. At least there's something growing in the ocean. But then, of course, this is just smothering everything underneath. So it's now a problem for sea turtles because it's just too dense. They can't deal with it anymore. It's shading huge parts of the ocean, and it's just getting worse and worse. And the people in these regions here, they simply don't know what to do with it. Those small islands simply don't have enough space to bury it. When it decays, it releases a lot of sulfur gas that really causes respiratory problems. Horrible for tourism, obviously. So yeah, I just wanted to mention that. And then my very first slide is the famous slide by Ribezil and Gattuso. I'm pretty sure it's the most shared slide ever in marine science. What it shows is what we're trying to achieve. So on the x-axis, we have our stressors that we test in lab experiments. We go from single stressor, just one impact, for example, temperature or ocean acidification, to two together, and then to multiple. And we always start in this little corner down here. We just work with one species in the beginning, and then eventually we might look at communities and ecosystems. But also the z-axis here is the time. What we actually want to understand is this very corner up here, the right upper hand corner. How do multiple drivers impact ecosystems after a long time, after they adapted to it? And in this slide here, they show that once we know this, we can then make informed decisions and informed policy about what to do. But we don't have that time. We're currently sitting maybe somewhere down here. So we know with multiple drivers, we're getting much, much better. Yeah, we're trying to work on ecosystems, but of course we don't have time to really look at what adaptation does, because that's over extremely long time scales that we would have to work with, especially with these longer lived organisms. So I personally think we do know enough. That doesn't mean that we should stop doing science or stop looking into things, but we can't wait. We can't afford to wait until we understand how everything is really working together before policy can do something. So we really need to urge that. Okay, that was it. Thank you.
Kia ora, Linn. Thank you for that. So we'll take questions now from Linn.
It depends a bit on where you are. So there are two things. It's one, it's larger plastic particles that break down over time. So all the rubbish eventually breaks down with wave action and UV light. or it's blown in there by wind, but then it's also small particles that enter the ocean as microparticles. And one of the main sources is synthetic fibers from clothes that we just wash, and there are filters. I hope one day every washing machine will have a filter, but you can buy them. I'm not selling them, but I'm promoting them. Car tires, abrasion from car tires is a huge issue. And then depending on how well each country deals with their rubbish, what they do with it, a lot of the rubbish, unfortunately, also enters the ocean, is just blown into the ocean. And then there's so much microplastic in the atmosphere, you know, all the synthetic fibers from carpet and whatnot that are blown into the atmosphere and also into the ocean. Yes, yes.
Yeah.
Thank you very much, Lynn. I just had a question about your graph of pH. So it looked like the Arctic in particular was higher pH, above 8, and then it looked like in the future projections it was going to go much lower than all the other regions. Was that right?
There is an impact of temperature, seawater temperature on pH.
Yeah, that one. Yeah, so I think green is the Arctic.
Yeah.
Yeah. So yes, I was just wondering what triggers that. So it's a temperature effect primarily, is that right?
Yeah, I'm pretty sure that in this graph, they also put the expected temperature change in there. I would expect that they have done that. So that might be. Kim, do you know more about this? Or that's...
Yeah, basically.
Yeah.
It's the solubility in cold water, but it's also the changing ice cover as well. So as you decrease the ice cover, you're going to get the oceans being able to absorb more CO2.
Kia ora, Lynn. I've got a question.
Yep.
So is bacteria always there
Yeah.
to be able to respond to phytoplankton primary production?
Yeah, bacteria are always there, and they can obviously respond really quickly. So they have usually very, very high growth rates, and we find them everywhere in the water column. So they're not just in deeper waters, but usually in deeper waters where we have this accumulation of organic biomass, we tend to find more of them.
So with sort of sporadic or random events like volcanic eruptions and the likes that might fertilize the deep ocean, you know, not a lot of that primary production would end up in the geologic layer. Would it be mainly feeding if zooplankton can't respond?
Yeah, well, it really depends on how quickly some organisms also can move into those patches of larger phytoplankton biomass. So that really depends on how quickly the grazers can respond to it and where you are and at what time of the year it happens. So how fast the growth rates are, then depending on temperature and these kind of things. But bacteria are always there, and they're, yeah.
So that figure of 1% sort of ending up in
that's really
geologic.
And that's a rough figure, and really simplification. Obviously, you have a huge difference there. Are there any questions from the Zoom people? Okay. Yeah.
So I think there are people who think this is just a technical problem that we can solve with geoengineering. And listening to you talk, the amount of things that are unknown and the complexities of the system seem to make that geoengineering idea seem just completely fantastical. But I wonder what you think about that.
Yeah, this is a tough one. I think the main worry I have about geoengineering is if we do something that we can't reverse quickly. So I think that's the main point when we're thinking about, you know, shooting things up into space that shade sunlight. Then you think, well, in 20 years we might not think that's such a good idea. I don't know. Yeah. So we really need, if we do something, we really need to make sure that it's something that we can control and that we can turn off. And I don't know what that would be, to be honest. I mean, ocean fertilization has been brought forward again and again as an idea for geoengineering. And as I just showed with the sargassum bloom, you can, you know, within pretty much no time, create these massive blooms and create this huge amount of biomass. But then what happens with it? I mean, that will, you know, you will have massive oxygen minimum zones underneath that. What happens to the ecosystem? So yeah, it's a bit overwhelming and frustrating thinking about it. I don't think that geoengineering is the solution. On the other hand, I don't think we have enough time to do, and we don't see the signs that we are willing to do something else. So at some point, we probably are going to be desperate, so desperate that we do something. Yeah, it's I don't know. I don't want to sound too desperate, but the best thing we can do is really reduce emissions. That's really the safest way to change things. And not only for carbon, you know, it would change so much for the better. Thank you.